Now we move to our last speaker, Dr. Rohimah Mohamud. Hello, Dr. Rohimah Mohamud. Can you hear me?
Hello.
How are you?
I'm good, thank you. Yourself?
Yeah, fine.
Oh.
Okay. Okay, ladies and gentlemen, before we start, I will give brief biography of Dr. Rohimah Mohamud. She is currently a lecturer at School of Medical Sciences, Health Campus, University Science, Malaysia. She completed her master degree at USM, Malaysia in vaccinology and continued her Doctor of Philosophy in Immunology at Monash University, Victoria, Australia. Now she will talk about update on COVID-19 innovation, including detection and vaccine. Please welcome Dr. Rohimah Mohamud.
Assalamualaikum and hello everyone. First of all, I would like to welcome everyone to this 8th Asian Academic Society International Conference, and I would like to thank and congratulate all the keynote speakers that has already delivered their excellent talk. And today I'm going to give a talk on the update on COVID-19 innovation, detection and vaccine development. Okay, inshallah. So this is the outline of the talk. I would briefly introduce my research group and the current research that leading by my group on COVID-19. Then we go to the effects of COVID-19 pandemic to the health areas, and then we go to the recent development of COVID-19 detection technique as well as the vaccine. As an introduction to my research group, I divided the research into the basic and fundamental research, and this is what I'm going to focus on the development of nanovaccine. For this one, we focus on the therapeutics as well as on the diagnostic. For the fundamental research, we intend to know why, what, and how tumor necrosis factor 2 mediate our immune responses towards these diseases. And we are also investigating what are the effects of all of these nanoparticles on the TNF-alpha cells and how they can regulate our immune system. When it goes into our body, it gives an effect. So that is what we are focusing on. And as you can see on the screen, there are different types of nanoparticles that we are focusing on. So we can see that polystyrene nanoparticles, silica, iron oxide, and etc. So when all of these nanoparticles goes inside our body, it has the different effect depending on the different characterization of nanoparticles. Okay, I give one example. If we use big particles such as like 0.5 micron particles, so it will target the macrophages. Okay, that is more to innate immune response. But let's say if we use 50 nanometer particles, or we call that nanoparticles, so it will target the dendritic cells. So we know that in vaccine developments, dendritic cells are important cells that is bridging between innate immune response as well as adaptive immune response. So these are the what happened in our lab basically. We just actually secured a funding by Asian University Network or SIDNet JICA for the special program for research for COVID-19 that is we call SBRSAC, and the project on the COVID-19 is on the ultra sensitive compact immunoassay system expressed biochecker using fluorescent Janus particles for the rapid detection of SARS-CoV-2. So for this one, we have the collaboration with Tohoku University, and the project started this month, actually. No, I mean last month. So they will post their nanoparticles to us. So this is their nanoparticles, and this is for the diagnostic purposes. And we will attach our specific ligand to the surface of these particles for the further assessment. But because of the COVID-19 pandemic and then the cases in Malaysia is increasing, they have to postpone the posting of these particles. So it may delay the process a little bit. So we're hoping that we can get these particles maybe early next year. We have some delays, so we expected to get the particles. We expected to get this the particle next year, inshallah. Okay, this is the effect of COVID-19 pandemic to health areas. I'm not going to go into details. We may refer to this report. And 2016 was considered as the start of year for SDG after 2015 MDG data, which published in 2017. So the report present the data for Asian and also for Asian member states So the data were gathered and compiled by the National Statistical System as well as National Statistical Offices in the Asia region from 2016 to 2018. So if you want to know more about this, we may refer to this one. This is the page 5 of the SDG report. We can see that Asian member states are facing issues and challenging in the collecting the data and etc. And this problem are worsened by the COVID-19 pandemic. Okay, in spite of the issues and challenges based on the SDG report that I show you, we are working hard to keep everyone going forward in achieving the sustainable nature. And besides that, there is a well-established health system support by other countries as showing in this slide. I'm not going to talk more on the effect of health post-COVID-19. So now we go to the virus detection. And here you can see that there are different types of virus detection technique. We can see that PCR, RT-PCR, we have the TM, and we have the nano biosensor, and etc. The current detection method for COVID-19 are the molecular and serological method, as you can see here. So these two methods are the main method to detect the virus. So if the individual get the positive, they have to do first they get the swab and then they perform the molecular methods. And second, they take the blood and do all the serological methods. These are the comparison of the molecular methods based on the sample, accuracy, time, professional skills, need, POCT, availability, cost, and high throughput. So we can see here the differences between these three, and all of these three methods use RNA sample. And in terms of the accuracy, all of these methods have high accuracy. Okay, for the times, we can see that RT-PCR, it takes more time compared to others. And all of these methods are limited in the term of availability, and the price are, yes, expensive. And then we can see the high throughput. That is the use of automated equipment to rapidly test thousands to millions of samples. And this is very important to test millions of COVID-19 samples, especially by using the RT-PCR. Okay, remember we have the what we call molecular methods, and another one is serological method. So this one, we have the lateral flow assay and ELISA. Okay, for the samples, all of this using the blood. So it's more to detection of antibody or antigen. And then for accuracy, it is from low to moderate. And the cost is a little bit lower compared to the molecular methods. So for the detection SARS-CoV at the early infectious stage by a rapid and accurate diagnostic method, that is important to save thousands of lives. So now I'm going to the other point of my talk. So this is the important thing. Go to the next slide, please. So which is on the vaccine development. Okay, currently there are 9 vaccine candidates in phase 3 of the clinical evaluations, and with the different vaccine platforms. And I'm going to show you there are 5 vaccine platforms. So here we can see that this is what we call viral vector vaccine. So as you can see on the top of the slide, that is number 1, viral vector vaccine. It involves, maybe it can be like replicating or non-replicating virus engineered to carry the gene. So most of the gene from the SARS-CoV, they focus on the spike protein. So the gene from the spike protein is inserted into the viral vector, and this candidate vaccine will be inserted into the cells. And you can see the other processes. This is another platform. We call that DNA vaccine. So as you can see here, the number 2 over there, DNA vaccine, it involves DNA that encodes the target antigens. So as I mentioned, the surface protein gene inserted into the plasmid. So you can see that the plasmid carry the specific genes for the SARS-CoV surface protein and then goes into the cells. So basically people do electroporation and etc. Next platform is the RNA vaccine. So as you can see there, number 3 please, the messenger RNA or mRNA encoding SARS-CoV surface protein. So just now we have the DNA vaccine that using the plasmid, but this one is they use RNA RNA. Yeah, because RNA just insert into the cells, they can basically self-amplify inside the cells in some of the RNA vaccine system. Okay, I forgot to mention.
1 minute left.
1 minute left. Okay,
Sorry.
the last one. Okay, go to
Okay.
number 4, please. That is the live attenuated vaccine. This one is attenuated, live attenuated or kill vaccine. People do kill either by the chemicals or irradiation. And then last one is the protein-based vaccine. So this is like a small protein from the SARS-CoV, and then basically people target or couple with the nanotechnology platform. So all of this vaccine is to evoke the immune system. Okay, so we've gone through all 5 different vaccine platforms. Okay, can we go to next slide? As I mentioned, we have like different vaccine platform. We have inactivate vaccine. We have the what we call recombinant viral vaccine. You just click, that's fine. And then we have the protein-based vaccine and mRNA vaccine. So all of these 9 vaccine that is already at the phase 3 clinical evaluation. That's ready to be what we call commercialized. Okay, these are the example. I list down all 9 vaccine. You just can keep going. Just keep going. Okay, yes. Okay, that's one. Okay, yep, all 9 vaccines. Okay, this is the conclusion. What is the direction for the future research? Ongoing progress of candidate vaccine through the preclinical and clinical studies. Yeah, this is a must. This and ongoing detailed characterization of the immunopathologies of the COVID-19. Implementation of large-scale post-marketing surveillance system to monitor SARS-CoV vaccine safety. And lastly, strengthen the vaccine development using a pandemic paradigm with the overlapping phases to shorten the development time. So because we have the pandemic and then the vaccine development, it cannot be using the traditional way. It has to be the pandemic paradigm. It means that it is overlapping. The phases is overlapping and we have a very shorter time. Okay, so lastly, I would end my presentation. Okay, before I end my presentation, I pray that all of us to stay healthy, practice social distancing, wear face masks when possible, and avoid crowded areas as the preventive measures against the COVID-19 coronavirus. So with that, I thank you for your attention and I welcome any new collaborator to our group. With that, I thank you.
Okay. Thank you very much, Dr. Rahimah Mohammed, for the presentation. It was interesting topic that we're still waiting for the availability of vaccine.